Hey, what's going on YouTube? Today we're going to learn about Paxlovid. It's a two-drug combination used for COVID-19 from mild to moderate COVID-19. And I'm going to go over the mechanisms and kind of make it more beginner friendly so that you can kind of understand and look at the mechanisms from a biological sense. Okay? So, meaning it's two-drug combination. So it's going to be nirmatrelvir and ritonavir, and I'll tell you why those are important. Okay. So basically, we start right here. We have SARS-CoV, which is this big red spike protein that we refer to it. And then we have our cell membrane at the top. And you can see it as ACE2, which is basically our channel. You can imagine it as a door. So SARS-CoV has these hooks, and these hooks come in and they grab the door, which allow them to just come right on into the cell. And unfortunately, when they come into the cell, they're kind of like any insect or creature. They have to excrement. They have to lay their eggs, so on and so forth. So RNA genome actually gets released. Now, this RNA genome we'll refer to as ssRNA genome, and it's going to be important. So let's go to our next step. Because it's RNA already, there is the RNA translation step, okay? So it's inside the cell. That means it's inside the home, and it can use its machinery. It can use the items in that house. So it's going to use the host ribosomes. And the ribosomes are the building factories. They're going to be able to read this ssRNA and then build long chain of proteins. And this chain we refer to in this scenario as PP or polyprotein, right? And there's two versions of these. They're going to be the PPA1A and the PP1AB, okay? PP1AB is the longer chain, okay? And the 1A and the 1AB is just referring to kind of like a page in a book. So the ORF1A and the ORF1AB, they're just where this ribosome does this movement with the letters, right? And it does something called a frame shift. We don't have to go into that specifically, okay? So with these polyproteins, the two chains, they contain about 16 tools in there, not for non-structural proteins. And this is the long list right here. You see NSP1 to NSP16. And NSP stands for non-structural protein. Okay, they aren't doing anything yet, right? But they will be very important when they're all there. Okay, so what happens is there is something called MPRO. Okay, and this allows it to cleave the polyprotein, the PP proteins that we talked about earlier. Okay, this is just a main protease, just cuts. Cleaving is cutting, right? And then once it does that, it produces the NSPs. And the NSPs together, they make the replication transcription complex, also known as the RTC, right? And this is the machinery that allows the virus to copy itself, right? So it needs all this to work, okay? So the idea that we're going to come across is, okay, we need to stop this from happening. So how do we do that? So what if we prevent the environment or MPRO to be unable to cut? So that's exactly how we're approaching it. So nirmatrelvir, what it does, it binds to the active site of the MPRO. It forms a covalent bond at the very specific location called, cis 145, and binds to it, and it's no longer in use. And then you may be asking, okay, so what's stopping it? Why is this two mechanisms, right? Well, we have um ritonavir, right? That blocks CYP3A4, and CYP3A4 is a liver enzyme that basically goes around and breaks down the foreign molecules. right? So the liver is doing its job, right? It's going to try to get rid of nirmatrelvir. So we have to basically stop it for a bit in order to combat, um, in a sense, the replication of this virus. Okay? And once that's done, because the replication is halted, you have all these uncleaved, uncut polyprotein chains just floating around in the cell, right? But, the RTC can't assemble and the replication can occur. So then you don't have any more viruses. And then in time, we hope that the adaptive immunity can mount an effective response to basically clear the remaining viruses. And that's pretty much the mechanism. Thanks for watching. If you liked it, please subscribe or leave suggestions in the comments. Take care.